Yesterday we had a lot of great discussions because the meeting was just the right size, just the right composition to encourage a lot of dialogue. So what I would like to do today is to present another technique that will allow us to look at dynamics, particularly large-scale dynamics in signaling proteins. I'm going to focus on protein kinase A because that's what we've done most of our work with. This is a collaboration with Susan's laboratory that started about almost 10 years ago, and with Jill Truella, who was at Los Alamos then and who is now at the University of Sydney. And she spends a little bit of her time in Utah, and I use her small angle scattering facilities there for local data. So, for a variety of reasons, pKa is a very good model system for looking at large-scale domain motions in protein kinases. It's dynamic. We know that it changes. Particularly, we've known for a long time that the dissociation of the C subunit is responsible for activation. So we've known for a long time that that's a dynamic part of the activation of this mechanism. But it's also a multi-domain protein, and these domains are very dynamic and interact with each other in ways that we are still trying to understand. So since many protein kinases, or most protein kinases, are also multi-subunit, multi-domain enzymes, this is a nice model system. We also have an incredible wealth of information, structural information from a variety of techniques. So this is a great opportunity to blend these methods and blend the data that we get from these different methods to get a very comprehensive picture of one of the prototypical protein kinases. So I'm going to spend today talking about the technique that we primarily use in our laboratory, small angle X-ray scattering. We can also use neutrons, and that gives us a little bit different type of information about the organization of subunits within a complex. I'm not going to say much more about that, except that it's a complementary technique. All the data, I think, that I'm going to be showing you today will be X-ray scattering data. So it's a solution technique, so that's one of its primary advantages. It's able to look at a wide range of molecular weights. In the literature, people have looked at molecules 6 kilodaltons and smaller, and megadalton are some of the sizes of some of the larger protein complexes that have been looked at. It's a complementary technique. It's not a technique that by itself is it tells the whole story, but it's particularly valuable when combined with X-ray crystallography, with NMR, with computational techniques, molecular dynamics. So I'd like to kind of keep that theme in mind, is how we can use this in combination with other techniques going forward. It's a low resolution technique. We don't get atomic level images or pictures, although we can incorporate atomic level models into our envelopes that we get from our scattering. We can develop three-dimensional models. The data is one-dimensional, but we can reconstruct three-dimensional models. We have to be careful about doing that. We can do that without making any assumptions about the size or shape. There's some techniques that have been developed, and I'll show you a little bit of data that use those. Or we can use data from other techniques, high-resolution data, to organize reasonable three-dimensional arrays or organizations of the domains and subunits using rigid body models. And this is still an area that's actively being developed.
Stupid question, when you do that to a molecule, it's not non-destructive testing.
The X-ray scattering?
Yeah.
Depends on the source of the X-rays. You can cook your sample. So at a synchrotron source, after a few seconds, your sample can be cooked. It falls out of solution. You know right away if you've done that. So another advantage is that it can be used for both rigid and flexible molecules. So it has a lot of advantages then in complement with techniques that can only be done with rigid models, or relatively rigid models. We can look at a variety of solution conditions. So we can look at changes in liquid concentration, pH, salt temperature. So we can access a variety of conditions, including what we consider might be physiological conditions. One of the reasons we started this was we were not able to see crystal structures of full-length protein kinase A, and so this became a complement to allow us to get information about these full-length models or molecules. The data collection is relatively rapid, so currently laboratory instruments have the capability of collecting data in minutes to hours. So typically, for most of the experiments that we do with protein kinase A, with a half an hour collection, we can get very good quality data at concentrations of around 2 mg per mil, 1 to 2 mg per mil. Using synchrotron sources, we can extend that into the millisecond domain, and so I'm going to show you a little bit of work that we've started to do for time resolved measurements where we get down into the millisecond time frame. And so we've used small angle scattering, both X-ray and neutron scattering, to look at a variety of different properties. We've looked at cyclic AMP-induced changes in structure and rearrangement of domains. We're using it now to understand a little bit more about the relative flexibility of different isoforms, and I'll show you a little bit of that data. The scattering is dependent upon the molecular weight squared, so it's an excellent technique for looking at association and dissociation, because your forward scattering, your scattering intensity, will be dependent upon whether or not you have a complex or whether that complex is dissociated. And we've used that to show that by addition of cyclic AMP to pKa, that we don't get full dissociation unless substrate is present. So that's provided us some new insights into the important functions of pKa. We're also beginning to look at a variety of mutations that change the interaction of the different domains within pKa. We can also look at modifications such as phosphorylation. So we have a variety of different conditions that we can look at, both in terms of the environment as well as the molecules. And we're also starting to be able to look more and more at kinetics, since the molecules are in solution and are moving, and that information is embedded in our data. So the basic information that you get from small angle scattering is size, shape, but we also get dynamic information, because when we shine X-rays on our samples, we are sampling all the conformers that are present there. We typically want to have a monodispersed system where all the particles are identical, although that's not necessarily the way we design some of our experiments, but typically that's one of the criteria is a monodispersed system. And as I just mentioned, we're looking at all the conformations, but also looking at all the orientations. So essentially, we're smearing out a lot of data, and that's one of the limitations of the model or the method. On the other hand, we are able, because of the fact that we're looking at all the conformations, if we have a very flexible molecule, we are looking at, in some form, the dynamics of that molecule. And if we do time-resolved measurements where we add a perturbant like ligand, like cyclic AMP, we can look at the structural changes that occur with time, if those are slow enough for us to access. So the basic scattering functions that we obtain from our experiments are the radius of gyration, which gives us an idea of the overall distribution of mass and the overall size of the molecule. We also get information from the forward scattering about the molecular weight, and this is proportional. This I0, the scattering extrapolated at zero angle, is proportional to the molecular weight squared, and it's proportional to the concentration of our molecule. We also get information about the molecular volume, and this will be important later on because this can tell us about the dynamics. You see a molecular volume that is much larger than we expect based upon the molecular weight. That's an indication that the molecule is occupying a larger volume, a lower density, but a larger volume than we might expect. Another important scattering function that we get is the length distribution curve, which tells us the interatomic distances within the particle between any two atoms. And this gives us an idea of the overall distribution of mass within our molecule. And then from the P of R, we also obtain the maximal end-to-end length of our molecule, Gmax. So you heard just a little bit yesterday about protein kinase A. I realized last night that we hadn't talked about enough to probably get everybody familiar with the jargon. So I will be using just a little bit of jargon, so I wanted to explain that as we go along here. These are the players, the major players that we're going to be talking about today. These are the R subunits, the regulatory subunits of protein kinase A. These are 3 of the 4. We're actually studying R1 data right now, but we don't have any data to show you on this. The domain organization consists of an N-terminal dimerization docking domain, which allows the R subunits to form homodimers. There's the inhibitory sequence. You heard a little bit about this yesterday. This is the region that resembles the substrate and fits into the catalytic site and inhibits the catalytic activity of the C subunit, and it also contributes to the binding energy. Site A is the first site of the AMP binding domain, and this, in combination with the inhibitory sequence, is the minimal critical sequence or structure to inhibit the C subunit and bind and dissociate in a cyclic AMP-dependent manner. There's an additional cyclic AMP binding site out towards the C-terminus that is also important, particularly with respect to cooperativity. With full-length protein, there's a cooperativity between these two sites, and it occurs through first binding of cyclic AMP to this site, which then allows cyclic AMP to bind to this site. One of the points I'd like to make here is that all the isoforms have this similar domain structure. The cyclic AMP domains are very homologous. The sequences are less homologous in the N-terminal region, but they're all about the same size, ranging in length from 374 amino acids to 420. So some of the terminology, the homodimer, we will refer to, or I'll refer to as the R2 complex without the C subunits. The holoenzyme are R subunits with C subunits. Typically, this would be the homodimer and 2 molecules of the C subunit. And then I will also mention, I think at least once, the heterodimer, which is a truncation where we have removed the dimerization docking domain, and we, in the experiment that I'm going to be showing you, it consists of a molecule that includes the inhibitory sequence all the way out to the end of the molecule. So here's some small angle scattering data. This is intensity on a log scale versus the angle. Here we've converted to a unit reciprocal angstroms. You'll see that the data look rather unremarkable, and you can tell that there's a large difference here in the shape of your molecule or size of your molecule, but we actually need to do an inverse Fourier transform to begin to, and other types of transforms to be able to extract some of our basic scattering functions. So this is the P of R plots, the length distribution curves for homodimers of the 3 different regulatory subunits, R1 alpha, R2 beta, and R2 alpha. You'll notice right away that they have different shapes. They have very different P of Rs, or at least R1 alpha does compared to R2 alpha and R2 beta. The end-to-end length, the maximal end-to-end length, is approximately 105, 110 angstroms for R1 alpha, whereas it's upwards of 180 to 200 angstroms for the R2 isoforms. In this experiment down below, we've added cyclic AMP to see what effect it has on the homodimer, and what you'll see is that it shows very little effect. So we can conclude from this that cyclic AMP binding to the molecules has a local change, but doesn't cause a global change in these molecules. Or the global change is rather small. However, in this set of experiments, we see that there is a large change in some of these structures induced by binding of the catalytic subunit to the homodimers. And this is isoform-specific, so again we have some scattering data up above here, which is hard to understand what is going on here, at least for most people. I think someone like Jeff Biol, who's looked at these things for many years, could tell you a little bit more than I can. So here are the P of R's for these different scattering data sets. Here we have homodimers, and we have the two R2 subunits, and then here we have holoenzymes of R2 alpha and R2 beta. And what you'll see is that even though the homodimers for R2 alpha and R2 beta are relatively similar, at least in their overall length, there is a significant contraction of the structure with R2 beta, whereas R2 alpha maintains that extended conformation. These were experiments done by Don Vigil, and one of the experiments he did was to see whether he could recapitulate this contracted structure by making a chimeric R2 that consisted of the R2 linker within the R2 beta structure. And he was able to get partially of that, but not completely. So that is information that indicates that this linker region is at least partially responsible for the difference between these two isoforms in their domain change in the presence of the C subunit. This more contracted structure suggests that the R1 alpha isoform is a more rigid structure, whereas the more extended structures are likely to be more extended. And we can support that with a little bit more data. We can easily build rigid body models for R1 alpha by taking crystal structures of the cyclic nucleotide binding domains and the high-resolution structure of the dimerization domain, and sort of fudging a little bit by making dummy volumes for the volume occupied by the linker region, which we don't have any structural information for. But we're able to use various rigid body modeling techniques to build a structure that looks like a Y for the homodimer. Doing this with the R2, in this case we were using both R2 alpha and R2 beta. I was showing the R2 alpha, but the R2 beta were the same. Trying to do the same thing, we didn't get very good models. The models didn't fit our data very well. So we went instead to this ab initio model using a program called Gammon, and we get a dummy bead model here. And one of the noteworthy aspects of this model is, well, two things. One is it shows a very extended structure, but the other is that this model also has a volume that is significantly larger than we would expect based upon just the molecular volume or the molecular weight. It's about 5% larger than we would expect. Whereas our models, if we use Gammon to generate this, we get a model that looks very similar to the rigid body model, and it's only about 15% larger than the molecular weight. So that would suggest that these forms are very dynamic. The homodimers are very dynamic. So now I'd like to show you that we can also see dynamics by looking at cyclic AMP binding. This is just one experiment where we use an antagonist analog of cyclic AMP, and a form of cyclic AMP that binds to the regulatory subunits, but doesn't cause dissociation of the C subunit. Here we're using one of the heterodimer molecules, so this is lacking the dimerization docking domain. So we're just looking at the inhibitory domain and the A and B domains. And when we add RP cyclic AMPS, what we see is a contraction of the structure. This part of the P of R curve looks fairly similar, but we draw in some amount of volume closer in, to about 105. This experiment suggested that we might be able to do some time resolve studies, which we were interested in doing. There's a large enough change here, so that led to some experiments that are ongoing right now in collaboration with Hiro Tsuruta at the Stanford synchrotron source. He is an expert in stop flow measurements in small angle scattering. Most of the work that has been done so far has been looking at protein renaturation, but since we see a large enough change in our scattering profiles, we decided this would be interesting to try. So this is the beam at the station, which actually was just rebuilt this past year, and we're going to be going out there again in March. This is the stop flow instrument. It's a standard stop flow mixing device, and so we can mix cyclic AMP with our protein molecule. That can trigger then the shutter to open and the detector to start, and we can collect data at various increments and bin those, and then go back and do analysis. And so this is our first experiment. It's still a little rough, but we didn't know what really what the time frame was or the quality of our data. But I think we were encouraged by this, so we're going to go back and try to get a higher resolution time increments. So the time increment that we're using here is 255 milliseconds. So in the first, this scattering curve here on the top here represents the average scattering over the first 255 milliseconds after adding cyclic AMP. We have an RG that's about 40 angstroms, and the I0 is about 2. The relative forward scattering here is about 2. The next 250 milliseconds, or 255 milliseconds, we see a small but significant contraction of the structure, and this is the P of R. And I've plotted this just for convenience, assuming a Dmax of 120. It probably would actually shorten as I showed you already, but I wanted to not bias this too much. I wanted to see what happens out in this region. And then by the third increment here, another 255 milliseconds, we see a further contraction of about another angstrom. And then by this time, we are close to what we see during the rest. We actually collected for 20 seconds in this experiment, and I just show well, I don't show any data here, but the data, if I take a couple of points out about 5 seconds and longer, we get an RG that is very similar to our last curve. So the conclusion here is that everything happens within about 750 milliseconds. Again, we'd like to look at this at shorter time frames. The new detector at SSRL will have higher sensitivity. We'll probably use higher protein concentrations. We'll do a few other things so we can get into that time range. The other thing I'd like to point out is that the I0 hasn't changed here. So what we've done here is we've added cyclic AMP, and this was done with cyclic AMP rather than that analog. And what we're able to see here is that there is no change in the I0 value, so we're not seeing any dissociation during this time frame. What we're seeing is just a domain reorganization. Okay. I'm going to jump ahead. We've been looking at another protein called EPAC2, which is another cyclic nucleotide binding domain containing protein that functions as a guanine nucleotide exchange factor. The cyclic nucleotide binding domains are in the N-terminus. The catalytic is here, and one of the reasons we're interested in this is that it's a single chain molecule and allows us to use another experimental approach to try to analyze the dynamic data. What we see is a very large conformational change when we add cyclic EMP. In this case, we see an extension. These are some of our ab initio models, and these are consistent with the recently published structure of part of the ECAP2 structure, where we see, or where the investigator saw an extension of the molecule. Now, they didn't look at the full-length molecule, and they also had GRAP1, the other protein that binds, that's activated, the small g protein that's activated. What I'd like to point out is that the volume is even larger, and so we use the technique called Ensemble optimization method, which is a relatively new method for looking at flexible proteins. And the reason I would like to talk about this is I think there might be some interest in using this approach for more small angle scattering, but I think there's some important reasons for looking at this more carefully by computational experts. So the technique is essentially to specify in a molecule parts that are rigid body and those that are linkers, and you keep the high resolution structure as rigid bodies and then allow these flexible linkers to assume lots of different conformers. You generate a large library, typically on the order of 10,000 conformers, where these linker regions assume various allowable phi-psi angles, and then you use a genetic algorithm to select 20 structures, typically among those an ensemble of 20 structures. And so the idea here is that in solution, a flexible molecule will assume an ensemble of different structures. In this part of the calculation, the scattering curve for each of those conformers is calculated, and then in the second part, those are combined to create a composite scattering function, which you compare to your experimental data, and what you get in the case of EPAC is something that looks like this. These are the RGs here. This is the average RG for the entire library, and we see actually two populations, neither of which corresponds to the average of that entire composite library, that 10,000 member library. These are the Dmax values in the distribution here. We see a short distribution and a long distribution, and when we add cyclic EMP, what we see is a change in the distribution. In the case of the Dmax, here we see a new shoulder forming, and this region here moves significantly out to a longer length. Again, we don't see an average molecule here, an average Dmax that corresponds to what we'd expect from other measurements. So I think that's something that is worth looking at in more detail. This has only been looked at in one paper in the literature, so I think that the technique needs to be validated more. So I apologize for going over a little bit here, but I will take questions.
Sure, Dawn, take questions as I said, but I have a question for you. In the original I0 data, the scattering data, shouldn't extrapolation back to zero give you the molecular weight?
It should.
So I noticed that the idea of trying to figure out if there's some aggregates there or not, you should be able to tell directly from the data?
That's correct.
Okay.
That's correct. So normally what you have is some sort of standard that you calibrate your I0 for your instrument. So you either have a protein that you know is low pH, or you use water as your calibrant.
So if you think for sure you have a flexible protein, do you use a flexible protein that's already there? Or do you use
the I0 is independent of the shape or
right.
flexibility.
Yeah. I realize that in theory. I'm just wondering in practice which gives you better results with those types of experiments. So what about an instance where you have some molecular dynamics, but the equilibrium applies far to, like, say, the closed structure? what percentage can you pick up of a smaller structure? I mean, obviously, if you go into really fast outflow, then maybe you'll be able to see the kinetics of interconversion. But if you're in the steady state realm, is there some?
I don't. To be honest, we just really started to use this one particular method, this ensemble optimization method. So I don't know what its capability. I don't even know if it's real, to be honest. So I think that's one of the things I would like to do is to be able to validate the method using computational methods as well as, let's say, fluorescence methods or other. Or we can develop a hypothesis based upon this and then test it using another technique.
So in terms of, so the data collection is relatively facile. I presume then the analysis is the rate determining step.
That's correct, yeah.
Okay.
I'm just curious whether when you reconstructed the R2 and R1 outputs you added the view.
I don't think we did in those experiments. I didn't actually do that. They do, yeah. And I don't think we did in those experiments. I didn't do that analysis, so I'm not absolutely positive, but I don't think we imposed symmetry on that.
Could you give me your opinion on the calculation of the approaches that are based on the calculation of the scattering pattern from a model versus approaches that try to take the scattering pattern and reconstruct an envelope, the so-called ab initio envelopes?
So I've been talking about this with some of the postdocs from Jill Troella's laboratory, and their general approach, and this is one that I think is one that we use, although we haven't formalized this approach, is to use a variety of different shape reconstruction techniques, including ab initio. If they all tend to agree, then you feel much more comfortable that you have something consistent. If they are wildly different, let's say rigid body models are very different from ab initio, then you need to question either the reconstruction technique or the data or both. I don't know if that answers the question, but that's sort of, it's pretty ad hoc. Each protein is very different. Some are very easy to describe because they're very compact. Others are more difficult, particularly if they're flexible.
So these multi-domain proteins, even R1 versus R2, where your length of your linker is very different between the modules that we understand the structure. Has anybody or you tried to look at systematic alteration of the linker or making the linker more flexible by adding things like glycine or what have you to see if you get what you expect in terms of expansion or compression of the molecule?
So Dom did the experiments where he made the chimera, so he did swap those out. The other experiments that we have done have been to alter the residues in the linker region. So I didn't have a chance to show you, but with the DPAC experiments, these were done in collaboration with Xiaodong Chen at UTMB, and we were looking at several site-specific mutants where he predicts that if you change the structure of specific amino acids from a small group to a large side chain, that you would restrict that hinge motion, and that's exactly what we see. So it's possible to go in and do both, sort of surgical as well as sort of large-scale changes.
Don, where do you get the shape curve that's a composite of two or more conformations? Is there a way by virtue of viscosity or cross-linking or temperature to come closer to the individual states where they're slowed down enough that you could begin to see them? And then that would provide a means of ascertaining what this overall shape is between the multiple conformations.
Well, we're still looking at a time frame that's incredibly long compared to the time frame for the reorganization of domains. We might have a possibility of doing that with some of the stop flow measurements, but I think even adding something that would change the viscosity, we still have dynamics that were much faster, since these are done in half an hour. So
But when did you change the equilibrium such that the two conformational states going back and forth were different? You get a shape difference, and that might be a third parameter to use.
That's right, and that's what we're trying. That's actually our hypothesis underlying the cyclic AMP addition is that the cyclic AMP we're able to shift conformers to a new set, so we would change that equilibrium. So that's sort of our working hypothesis.
I was wondering if you could just comment briefly on what the status is of the ab initio methods. I assume those are the ones that are sort of the inverse Monte Carlo type things where you try to build something up that matches the scattering profile and if they could use for small calcium binding proteins.
Well, this Gammon is a simulated annealing program. There are different ones, and maybe Mike might be more familiar. He's going to be talking about this a little bit, but those are still a lot of different models out there. I kind of use the ones that are readily available to me. So this Terhune group has put a lot of their programs out for the community to use. But I would encourage anybody that is interested in participating. We'd be glad to provide lots of small angle scattering data to try. But there are different techniques. I know that the group at Los Alamos, when Michael was there, had various techniques that they used. And some of those are proprietary. They're not generally available, which makes them hard to compare to other techniques.
One last question.
Can you comment a little bit on your hypothesis about the cyclic AMP binding? Why do you see a more compact complex instead of seeing dissociation? I mean, it's kind of opposite thing that you would expect, right?
So, you mean with the R2 beta, where we have
the heterodyne, when you have a domain and you put the cyclic AMP and you see more smaller.
or with the RP. So that was actually a holoenzyme structure, the C subunit, and I think that's consistent with the X-ray crystal structure as well.
The RP binds but doesn't dissociate.
No, that was cyclic AMP with A domain.
No, I didn't show anything with just the A domain.
There was one question over here. I realized I was ignoring the small side of the audience.
Do you believe that you can see more than intermediates when you do the stop-flow measurement? Because the resolution obtained is not really very high in terms of determining things. So the question is if you can determine essentially beginning and end only, what is really the purpose of doing there?
Well, you're right. My goal or my dream, I guess, would be to see a conformational intermediate that you wouldn't normally see under equilibrium conditions. I guess the other thing that I think would be valuable, particularly for the people doing molecular dynamics, long-range molecular dynamics, would be to just follow what the time change is. Do you go have a fast and slow changes?
But that kind of information can be obtained by other methods.
That's right.
Just taking it. They say it's the intermediates that you cannot obtain.
Right.
So I think at this point we should probably end up, except I need to make a comment. Those of you who know me, I